My name is Oliver Fien. I'm the director of the West Coast Metabolomics Center. Welcome everyone to this month's West Coast Metabolomics Center online seminar, where we have invited Professor Maria Veiga da Cunha. She is also chercheuse qualifiée at the FNRS, which is Faculty of Pharmacy and Biomedical Sciences at the Institut de Douve at the Université Catholique de Louvain in Belgium, which is in Woluwe-Saint-Lambert. And she did her PhD at the biochemistry department at the University of Oxford from 1987 to 1990, before she then went to a postdoc position at the Instituto de Tecnologia e Química Biológica in Oeiras, Portugal. Yeah. I always have to think about, like, how would I pronounce this? Before she then went to the Université Catholique de Louvain, where she's working in metabolism and enzymology since 1992, including working on protein expression and purification, with a special interest in molecular identification of enzymes with applications to inborn errors of metabolism. So Professor da Cunha, therefore, will give us today her presentation about metabolite repair and everything that is related to it, specifically a story of metabolite repair and glycolysis, and the failure to eliminate a phosphorylate glucose analog strongly impairs glucose metabolism, and the impact on inborn errors of metabolism. So thank you so much, Maria, for coming to us remotely. We wish we would be able to welcome you in person next time. We will. So the floor is yours.
Okay, thank you. So I'm going to share my screen. So good morning, everybody, or good afternoon, or good evening, if you're closer to where I am. So Professor Fien, thank you for giving me the chance to talk to you about this story that I really like very much. And so what I would like to do today is begin by talking about enzyme specificity, or actually lack of enzyme specificity, and how this led us to develop this concept of metabolite repair, who is now making us have a different view, looking at intermediate metabolism in a different way. So I'd like to do that maybe in a more general way, and then focus more specifically in this story of metabolite repair in glycolysis, where we found that an analog of glucose 6-phosphate impairs glucose metabolism and explains the neutropenia in these two inborn errors of metabolism that I'll be talking to you about today. So about enzyme specificity. So what you can read in this reference book, which you probably all know, and I have it there in my shelf, it's that enzymes have a vastly greater degree of specificity with respect to the identities of both their substrates, reactants, and their products than do chemical catalysis. So what this means is that enzymatic reactions rarely have side products. Well, this is probably not completely true. It's actually not true at all. So fortunately, enzymes are quite specific for their substrate, but nevertheless, if in the cellular milieu we have metabolites that are normal metabolites and that are structurally similar to the normal substrate of this enzyme, then the enzyme will have a side reaction on this normal metabolite and will produce what we call a non-classical or an abnormal metabolite. Now, how come usually when you look at cells, you don't see these non-classical or abnormal metabolites? Well, it's either because they are destroyed, because they are unstable or whatever, they are destroyed, or because we have dedicated enzymes that we call repair enzymes that are going to act specifically on these non-classical metabolites and convert them back to a normal metabolite, which is part of which is an intermediate of the normal metabolic pathways that we know of. And these ones, of course, are not intermediates of those normal pathways that we know of. Now, these repair enzymes are often extremely conserved, which suggests that they are actually quite essential. And in fact, if you have a mutation in a gene that codes for one of those repair enzymes, what you see is that these non-classical metabolites, these abnormal metabolites, are going to accumulate. And then this could lead maybe to some toxic effects, even a disease. And when we have a disease, we're now beginning to call this disease inborn errors of metabolite repair. So now I'd like to tell you how we actually came across and started to work on these enzymes of metabolite repair. So this dates back to 2004, and that's when we discovered the gene that it's mutated in L2-hydroxyglutaric aciduria. So L2-hydroxyglutaric aciduria is a neurometabolic disorder where the patients have a progressive destruction of the white matter, and this leads to a severe mental retardation. So these patients, they accumulate L2-hydroxyglutarate in brain, in their physiological liquids, which is in part excreted in urine. And so the gene that is mutated codes for this enzyme L2-hydroxyglutarate dehydrogenase. And so L2-hydroxyglutarate dehydrogenase is a mitochondrial enzyme, it's FAD dependent, and it converts L2-hydroxyglutarate to alpha-ketoglutarate. And so when this enzyme is deficient, when there is a mutation, then you accumulate quite a lot of L2-hydroxyglutarate. And it's when we were wanting to find the origin of this 2-hydroxyglutarate, because it is not, it's not the normal intermediate of the pathways that we all know. So it's when we were looking to find what was the origin of L2-hydroxyglutarate that we realized that it was the origin of L2-hydroxyglutarate, was a side reaction of this enzyme, which is mitochondrial L-malate dehydrogenase. It's also therefore a mitochondrial enzyme, which converts oxaloacetate to L-malate. So this enzyme will have a side reaction on alpha-ketoglutarate, which, as you can see from what the structures I've drawn here, it has just one carbon more than oxaloacetate, so it's very similar to oxaloacetate, but alpha-ketoglutarate is quite abundant in the mitochondria, and so malate dehydrogenase is going to act on it and form L2-hydroxyglutarate. So the difference in catalytic efficiency between these two reactions is 10 million, which is huge. However, because this enzyme is so abundant, this side activity, when you don't have L2-hydroxyglutarate, is sufficient for the liver of a non-normal adult to make 1.2 grams of L2-hydroxyglutarate per day. So this side reaction will make 1.2 grams of L2-hydroxyglutarate per day. So you can imagine that if you don't have L2-hydroxyglutarate dehydrogenase, the enzyme that detoxifies this, you're going to accumulate quite a lot of that. And then, of course, you have the toxicity and the disease. Okay, so now keep this in mind, these ideas of metabolite repair, because we're going to need them later on in my talk. So now I'd like to switch gears. So I wanted to now begin to talk about the two neutropenias. But before I do that, I need to remind you a little bit about some basic concepts of glucose metabolism in liver. So the reason I want to do that is because one of the proteins that it's mutated in these neutropenias is the glucose 6-phosphate transporter of the endoplasmic reticulum. So the glucose 6-phosphate transporter on the endoplasmic reticulum also has a role in the liver to maintain blood glucose homeostasis. And so what happens is when we fast, glycogen is degraded to glucose 6-phosphate, and the neoglucogenesis is also going to produce glucose 6-phosphate. So this glucose 6-phosphate will get inside the endoplasmic reticulum via the glucose 6-phosphate transporter. And there we have a dedicated phosphatase, which is the glucose 6-phosphatase, that is going to dephosphorylate the glucose 6-phosphate, produce glucose, which can get back into the leave the endoplasmic reticulum, leave the hepatocyte, get into the blood to produce glucose, when we are fasting. Okay, so the function of these two proteins is so important that if you are mutated in G6PC1, the glucose 6-phosphatase, which is expressed in liver, in kidney, and intestinal mucosa, you have a disease which is called glycogen storage disease type 1A. So these patients, they have a big liver because it's full of glycogen. They suffer from hypoglycemia and lactic acidosis. Now, if you are deficient in the glucose 6-phosphate transporter of the endoplasmic reticulum, which, unlike G6PC1, is expressed in all tissues, you have a different disease, which is called glycogen storage disease type 1B. So these patients have exactly the same symptoms as the glycogen storage disease type 1A patients, but very surprisingly, they also have neutropenia. Now, if you are mutated in G6PC3, which is another phosphatase of the endoplasmic reticulum, which is from the same family as G6PC1, they have about 40% identity, and which is called ubiquitous glucose 6-phosphatase. It's also present, expressed in all tissues. There you have a different disease, which is congenital neutropenia type 4. And in this disease, these patients, they have no metabolic symptoms, but they have this neutropenia, which is associated with cardiac and urogenital malformations. And then you also see they have very prominent veins in their torso when you look at them. Okay, so this is the situation when we begin to study the origin of this neutropenia. So we were puzzled by the fact that these two diseases, when you are deficient in G6PD and you are deficient in G6PD, you do have a neutropenia. And the explanation that was given in the literature for that, which I'm just going to tell you now, we thought it was surprising. So we didn't think that was the good explanation. And so therefore we wanted to work on that and try to find it out. So this is the explanation that was given for the origin of this neutropenia. So now here we have a neutrophil. And so glucose is transported inside the neutrophil, and it's phosphorylated by exokinases or ADP glucokinases to glucose 6-phosphate. So this glucose 6-phosphate is going to feed into glycolysis to produce ATP for cell survival. It will feed into the pentose phosphate pathway to produce NADPH that will be used for the respiratory burst. And then it will also serve to make UDP glucose for the production of glycans for protein glycosylation. But so what was said was that it would also get inside the endoplasmic reticulum by the glucose 6-phosphate transporter, and there it would be dephosphorylated, the glucose 6-phosphate would be dephosphorylated by this phosphatase G6PC3 back to glucose. So that if the patients were mutated in, the patients or the mice were mutated in G6PD or G6PC3, what would happen, what was seen was that there was less glucose 6-phosphate, and therefore less ATP, less NADPH, and less glycans and problems with protein glycosylation. And so the end result was that there was endoplasmic reticulum stress in these neutrophils, glycosylation defects, reduced respiratory burst and phagocytosis, and an increased apoptosis, therefore the neutropenia. However, I think you will agree with me that if we don't have the glucose 6-phosphate transporter, or if we don't have G6PC3, you don't really expect to have less glucose 6-phosphate. You expect to have more glucose 6-phosphate, and therefore more ATP, more NADPH, and more, and not a problem with the protein glycosylation. And that's not what was seen. So the question that we asked ourselves was, is G6PC3 really a glucose 6-phosphatase? So how did we address this problem? So what we did was we cloned we cloned G6PC1, so the glucose 6-phosphatase. Here we knew what the function was, and we cloned G6PC3 into plasmids that would allow us to overexpress these proteins into AGK293 T cells. So we transfected the cells, and then we isolated the membranes, hoping that the membranes would be enriched in either functional G6PC1 and functional G6PC3. But we could not purify these proteins because these proteins are embedded in the membrane. And so and then what we wanted to do with these things was to go and measure the phosphatase activity on a series of phosphoric esters, right? But so we wanted to make sure that the dephosphorylation that we were measuring was really due to one or the other of these phosphatases. So to make sure that this was the case, what we also did is we created some catalytically crippled mutants. So we mutated this histidine here, which is the catalytic histidine, to have an inactive enzyme. And so we compared the activity of the membranes isolated from transfected cells with the wild type G6PC1 to the mutated G6PC1, and then the wild type G6PC3 with the mutated G6PC3. So these two here, the mutations were just a base phosphatase activity that was nothing to do with the phosphatase of G6PC1, the phosphatase activity of G6PC1 or G6PC3. So then we used these membranes to measure the phosphatase activity of G6PC1 and G6PC3 on a series of different phosphoric esters. So the first thing that we can see is actually that G6PC3 is really not at all a good glucose 6-phosphatase. It's about 10-fold less active than the real glucose 6-phosphatase, which is the glucose 6-phosphatase found in liver. So the other thing that you can see is that G6PC1, so the glucose 6-phosphatase, is a much more specific enzyme than is the G6PC3 that acts on a series of, so it dephosphorylates a variety of phosphoric esters. So here you can see that G6PC1 also dephosphorylates mannose 6-phosphate, but actually the glucose 6-phosphate transporter does not transport mannose 6-phosphate. So once it's inside the endoplasmic reticulum, G6PC1 is actually quite specific. So, so for G6PC3 that we were interested in, we have all these potential substrates. So we got interested in the more, the beta substrates, which already had an activity that was higher than the glucose, the activity on glucose 6-phosphate. But for this, there to be a connection between the lack of dysphosphorylation of ribose 5-phosphate or ribitol 5-phosphate or even pyrophosphate, these ones, they had to be transported by the glucose 6-phosphate transporter, right? So we needed to find a way of measuring the transport. And so that's what we did here. So to do that, we prepared microsomes from rat skeletal muscle to be able to test the specificity of the glucose 6-phosphate transporter. So microsomes are vesicles of the endoplasmic reticulum. So we used rat skeletal muscle because it had no G6PC1. Remember, it was present in liver, kidney, and intestine, but it was quite rich in G6PC3. And we knew also that it had the glucose 6-phosphate transporter. So what we did was we took these vesicles and we incubated them with glucose 6-phosphate just to check that, you know, our way of measuring transport worked, because so we knew that G6PT was a glucose 6-phosphate transporter and we wanted to see if this worked. So if glucose 6-phosphate enters these vesicles via this glucose 6-phosphate transporter, then G6PC3, which has a little bit of activity on the glucose 6-phosphate, is going to produce inorganic phosphate. And this is what we are measuring here. That's the activity that we are measuring here. So we see a bit of activity on glucose 6-phosphate, but we know G6PC3 is not a very good glucose 6-phosphatase, okay? So then to make sure that we were really measuring the transport via G6PT, we used a specific inhibitor of G6PT. And in that case, so we do exactly the same experiment. And in that case, you can see that the activity, the G6PC3 activity is decreased quite a bit. And so this tells us indeed that the glucose 6-phosphate transporter is being transported by this specific G6PT. And so the question was, are the other good substrates of G6PC3 also transported by G6PT? So we did exactly the same type of experiments on the best substrates, so ribose 5-phosphate, ribitol 5-phosphate, and inorganic pyrophosphate. And we did them without the without inhibitor or with inhibitor. And you can see that in any case, the inhibitor would inhibit the phosphatase activity, the G6PC3 activity on these substrates. And so this was telling us that none of these good substrates of G6PC3 were substrates of the glucose 6-phosphate transporter. So at this stage, we were actually quite stuck because, as I said, none of the good substrates of G6PC3 was transported by the glucose 6-phosphate transporter. But however, these experiments nevertheless had told us something, because they made us suspect that the physiological substrate of G6PC3 was probably very similar to glucose 6-phosphate, so that it could be transported by the glucose 6-phosphate transporter. But the problem was that we already tested all the phosphoric esters that made sense. So it's now here that this idea of metabolite repair and this idea of enzymes not being very specific came to our rescue. And so the question that we asked ourselves was, could the substrate of G6PT or G6PC3 be an abnormal metabolite? So could it be the product from an enzymatic side reaction? So in other words, could glucose phosphorylating enzymes phosphorylate a glucose lookalike to make an abnormal glucose 6-phosphate lookalike? And so, as you know, the enzymes that phosphorylate glucose that are more abundant in our cells are exokinases. So it's here that we went to look again at, to go back and look again at the substrate specificity of exokinases. And then we found this paper, which is a 70-year-old paper that was written by Alberto Sols and Robert Crane, where they studied the substrate specificity of brain exokinase, which is exokinase 1. And here, what we saw was that, so this is the activity on glucose, okay? So exokinase is phosphorylating glucose on glucose 6-phosphate, but we saw that it also was able to phosphorylate 1,5-sorbitan, about 3,000-fold less well. But nevertheless, it was able to phosphorylate it, and that's what we expect from side activities. So 1,5-sorbitan is 1,5-anhydroglucitol, which is 1-deoxyglucose. So it's glucose with one oxygen less on carbon 1. So as you can see, it's very similar to glucose. Structurally, it's very similar to glucose. So this 1,5-anhydroglucitol, this 1-deoxyglucose, is a polyol. It's probably one of the most abundant polyols that we have in our blood. Mice have it, and it comes from food. So our hypothesis was the following. So 1,5-anhydroglucitol, which is present in our blood at about 150 micromolar, so about 30-40-fold less than glucose. And then I'm showing you here again, when you compare these two, glucose and 1,5-anhydroglucitol, you see there is just one oxygen less on this first carbon. So the hypothesis was that 1,5-anhydroglucitol would get in the neutrophils, and then by a side reaction of the exokinases, would be also phosphorylated, and we would have 1,5-anhydroglucitol 6-phosphate. And then the idea, what we thought, was that 1,5-anhydroglucitol 6-phosphate would be transported by this glucose 6-phosphate transporter, and there it would be dephosphorylated by G6PC3, which was therefore preventing its accumulation. So this would be our abnormal metabolite, and this would be our repair system. So that if the cells were deficient in the glucose 6-phosphate transporter or in G6PC3, then we would accumulate quite a lot of 1,5-anhydroglucitol 6-phosphate due to this side reaction. Now, okay, you make a lot of 1,5-anhydroglucitol 6-phosphate, but then why would this be a problem? Well, it's because 1,5-anhydroglucitol 6-phosphate is a good inhibitor of the exokinases, which are the enzymes that convert glucose to glucose 6-phosphate. So we were kind of blocking the metabolism of glucose in the neutrophils. So what you have to know is that exokinases are physiologically inhibited by glucose 6-phosphate. So they have a binding site for glucose 6-phosphate, which is the same binding site that binds ATP. And therefore, what we're probably seeing here is, again, a kind of lack of specificity when we have a lot of 1,5-anhydroglucitol 6-phosphate, which is an analog of glucose 6-phosphate. This is going to bind to that site in exokinases and completely inhibit these exokinases. And so, if this was so, then what people were seeing in these neutrophils that I told you in the beginning, which was when the neutrophils from the patients or from the mice deficient in G6PD or G6PC3, what was described was that there was less glucose 6-phosphate, less ATP, less NADPH, less protein glycosylation, would now begin to make sense, okay? So if this was true, then G6PC3, if we go back to our in vitro system, should be a good substrate. 1,5-anhydroglucitol should be a good substrate of G6PC3. And that's exactly what we see here. It's a much better substrate than glucose 6-phosphate, and it's about more or less as good as the other ones that we have seen. And it is not a good substrate of G6PC1. So then the other thing was, is it transported by the glucose 6-phosphate transporter in those microsomes that I've told you before? So again, the answer is yes, because it behaves exactly like glucose 6-phosphate. So when we add the inhibitor, we see that we are inhibiting the G6PC3 activity, which is what we're measuring here, because 1,5-anhydroglucitol 6-phosphate cannot enter the vesicles and therefore cannot be dephosphorylated. So now, seeing all of this, what we did was we created a human cell model that was deficient in G6PT or deficient in G6PC3, and we incubated these cells with increasing concentrations of anhydroglucitol. So this is the physiological, more close to the physiological concentration of anhydroglucitol. And you see that only when there is no G6PT or G6PC3, the cells are going to accumulate anhydroglucitol 6-phosphate. So if you have more, then of course you'll have more accumulation of 1,5-anhydroglucitol 6-phosphate. And in the wild type cells, you don't see any accumulation, nor if the cells are incubated in the absence of anhydroglucitol. So we concluded that G6PC3 and G6PT collaborate to destroy 1,5-anhydroglucitol 6-phosphate. And then we wanted to look in a cell model that was closer to neutrophils. So we used immortalized neutrophil progenitors that were derived either from wild-type mice or mice that were deficient in G6PC3. So and then we incubated these immortalized neutrophils with increasing concentrations of anhydroglucitol, and we measured survival. And then what you can see is if there is no anhydroglucitol in the media, both neutrophils survive the same way. So this was after 48 hours. And when we began increasing the concentration of anhydroglucitol, then you can see that the neutrophils, and only those that are deficient in G6PC3, begin to die. So then what we did was we took this physiological concentration, this concentration of anhydroglucitol, 200 micromolar, so close to the physiological concentration, and then we incubated both the wild type and the G6PC3 knockout neutrophils for 24 hours. And we could see that over time, the neutrophils that had no G6PC3 would accumulate anhydroglucitol 6-phosphate to concentrations that were close to 3 millimolar. And so then we wanted to see what was the effect of accumulating these large amounts of anhydroglucitol 6-phosphate in the products in the intermediates of glycolysis. And so what you see is that very logically, in this case, when you accumulate anhydroglucitol 6-phosphate, you are decreasing the concentration of glucose 6-phosphate because you are blocking exokinase. And then if you look further down in glycolysis, you see that the same thing happens with fructose 1,6-bisphosphate. So then what we did here was we took blood from patients that were either deficient in G6PT or deficient in G6PC3, and we isolated the granulocytes, and then we put them in the LCMS, and we went to look to see if we could find nitroglycerol 6-phosphate, and we did the same thing for 3 controls. And what you see is that the concentrations, when we calculate the intracellular concentrations of nitroglycerol 6-phosphate in the neutrophils of these 2 type of these patients that come from these 2 different types of neutropenia, they accumulate also about 3, 4, 5, sometimes maybe even more nitroglycerol 6-phosphate, which is huge, particularly if you see that the inhibition constant of hexokinase 3, which is the main hexokinase in neutrophils, is 130 micromolar, the inhibition constant for 1,5-nitroglycerol 6-phosphate. So here we're talking about 30 times more nitroglycerol 6-phosphate than this value here. So I guess metabolism of glucose, the phosphorylation of glucose in these neutrophils must be not 0, but not far from it. And when you then go and look at nitroglycerol in the serum of both controls and patients, you see that the amounts are very similar. So we had understood the mechanism of this neutropenia we thought. So the next question that we asked ourselves was, can we treat these patients? Is there a simple way of lowering the concentration of 1,5-nitroglycerol that we have in blood? Because this could be a good way of maybe trying to treat these patients. If we don't have the metabolite where the side reaction is happening, then we have no product, and therefore this might be a good way of helping these patients. So I told you that 1,5-nitroglycerol comes from food, and so you could think, okay, well then let's stop eating foods that contain nitroglycerol, but basically you find it everywhere. So this was not a very good solution. And so when we were looking into that, we found that there was this new class of type 2 antidiabetics, which are inhibitors of Sglt2, which is the main glucose transporter in the kidneys. And so these inhibitors are often used to treat diabetes, because what they do is they cause a massive glucosuria. So you are preventing the glucose in the kidney filtrate to be reabsorbed into the circulation. And so they cause this massive glucosuria. And what was also described in the literature was that this lowered the plasma concentration of 1,5-nitroglycerol. So let's take a look at how this works. So, the blood is filtered in the kidney, and then in the renal filtrate, you are going to have a bunch of metabolites, and you'll have glucose, which I've shown here in green, and you also have nitroglycerol, which we have in blood, right? So the glucose is reabsorbed back into the circulation in the renal tubule by this transporter Sglt2. And then further down in the renal tubule, you have another transporter, which we think is Sglt5, which is reabsorbing 1,5-nitroglycerol. Now, this transporter is not specific for nitroglycerol. It transports mannose, fructose, glucose, but we think this is the transporter that is reabsorbing nitroglycerol and bringing it back into the circulation. So now, if we take these Sglt2 inhibitors, empagliflozin is one of them. There are 6 in the market. This is one of them, is the one we used. So you block the transport of Sglt2. And so what you're doing is you're blocking the reabsorption of glucose. So if you're diabetic, this is very useful because you can, like that, get rid of the extra glucose that you have in your blood. But if you are non-diabetic, this is also not a problem. You just have glucose in urine, but you don't get hypoglycemic or anything like that, because there's actually another transporter that also reabsorbs a bit of glucose. Well, but nevertheless, so what you do, you are enriching your renal filtrate in glucose. And by doing that, because remember, I told you that this is not a specific transporter for nitroglycerol, it also seems to transport glucose a little bit. You are preventing the nitroglycerol from getting back into the circulation. And this is why what was written in the literature was that these inhibitors caused a massive glucosuria and lowered the blood concentration of nitroglycerol. So what we first did was we tried this empagliflozin, so this Sglt2 inhibitor, in our mouse model deficient in G6PC3 to see if we would decrease the concentration of nitroglycerol in the blood. And so we gave to both wild type and G6PC3 deficient mice empagliflozin, so this Sglt2 inhibitor, for 2 weeks. And what we saw is that after 2 weeks, we had decreased the concentration of nitroglycerol in the blood of these mice by about 5-fold. So you see that the initial concentration here is 50 micromolar. So we have about 150 micromolar. So we were interested to get to levels closer to what we have. So we also fed the mice with nitroglycerol, and in that case, we managed to increase the concentration of nitroglycerol in the blood of these mice closer to the concentration that we have. And then we went to look at the neutrophils of these mice to see what was happening with nitroglycerol 6-phosphate. And that's what you see here. So we took the leukocytes of the mice, so the heterozygous mice, untreated heterozygous mice, or mice that were deficient in G6PC3. And you can see that the neutrophils of the mice deficient in this G6PC3, very logically, are going to accumulate nitroglycerol 6-phosphate. And these mice, also quite logically, they are neutropenic. So they have, here we're looking at granulocytes numbers, so neutrophil numbers. You can see that they have about half of what normal mice do have. So they are, you can see that they are neutropenic. Now, we treated the mice with empagliflozin, and after 2 weeks, we went to look at the leukocytes of these mice, and you can see that we had lowered quite a lot the amount of nitroglycerol 6-phosphate inside these neutrophils, and it's almost normal. It's not normal, but it's almost normal. And we had recovered neutrophil counts, so these mice also have more or less almost normalized their neutrophil counts. So they are not neutropenic anymore. And then the mice that were fed with nitroglycerol, they increased their concentration of nitroglycerol by about 3-fold compared to these ones. And these mice became profoundly neutropenic. So we are able to treat the mice, as you can see. So the next question was, can we use this actually to help these patients? Are we going to be able to treat these patients with the same Sglt2 inhibitor? So this was, I mean, I think we first started treating the first patient about a year and a half ago. Now there have been quite a few patients that have been treated, both patients with glycogen storage disease type 1b and patients deficient with G6PC3. In all cases, this has been beneficial. There's a variability in the response. Some respond better than others, but in general, this has been quite successful. And so what I'd like to do now is take you through the first patient that we treated to show you the actual results. So this patient was a lady, a 21-year-old lady, who's a patient of Dr. Saskia Wortmann in Austria, and she was the perfect candidate to be treated with empagliflozin because she had a long history of neutropenia and neutropenia-related issues. And so she had constant oral, anal, and urogenital lesions and infections. She had an overt inflammatory bowel disease with about 20 bloody and watery stools a day. So she actually had a bit of pain because it was so inflamed and so damaged that they had to section that out. And so she had constant abdominal pain. And then what we were doing before we found all this out to these patients to treat their neutropenia was they were receiving injections of GCSF, which is granulocyte colony stimulating factor. So it's a cytokine that stimulates the production of neutrophils. Okay, so in the case of these patients, these injections of GCSF did not seem to be able to control infections anymore. So she was a good candidate to begin with. And so what we could see, so she began to take the Sglt2 inhibitor. And what we could see when we looked at the concentration of anhydroglycitol in the plasma was that after 2 to 3 weeks, it had been decreased by about 3 times, threefold, but it was not zero. Okay, and so when we looked at anhydroglycitol 6-phosphate in the leukocytes, we saw exactly the same. We had reduced quite about 4 or 5-fold, more or less the same way as anhydroglycitol, but again, it was not zero. So if you look at my neutrophils or your neutrophils, we have very, very little, almost nothing. So they still had a significant amount of anhydroglycitol 6-phosphate. So we wondered if this was sufficient to improve the function of the neutrophils. So we wanted to look at the neutrophil function, and to do that, we looked at 2 things. We looked at protein glycosylation. So remember, I told you that anhydroglycitol 6-phosphate was inhibiting exokinases, decreasing the amount of glucose 6-phosphate. So we have less UDP glucose, and then these neutrophils had problems of protein glycosylation. So we wanted to see if we could improve protein glycosylation. So to do that, we looked at this protein LAMP2 in the neutrophils of our patients. And this protein is a protein that is heavily glycosylated. It has 15 glycosylation sites. And so it was a very good candidate to measure protein glycosylation. So when you take the neutrophils, you make an STS gel, and then you use antibodies against LAMP2 to detect LAMP2. So if you do that in controls, so in neutrophils from a control patient, LAMP2 migrates at about 115 kilodaltons. And then if you do that with the neutrophils from the patient before treatment, you really see that there is a big defect in protein glycosylation. So there's a large amount of LAMP2 that it's not properly glycosylated. It's under-glycosylated. And then after 28 and 50 days of treatment, you do the same thing, and you can see that you have indeed recovered most of the glycosylation, the protein glycosylation. So you have recovered this glycosylation defect. Even though these patients don't have, we didn't get the anhydroglycitol 6-phosphate down to zero, down to the same level that we have, we had improved at least quite a lot the protein glycosylation in the neutrophils of these patients. So the other thing we then looked at was the respiratory burst. So you know that neutrophils are there to produce, to make respiratory burst to kill bacteria. So this was a very important function, and we wanted to make sure that we had improved it. And so the question was, do we have enough NADPH oxidase? Can we produce enough NADPH for NADPH oxidase to act well, and so for the respiratory burst to happen? So, to do that, we measured the fluorescence of dihydroxyrhodamine. So dihydroxyrhodamine gets inside the neutrophils, and then when it's oxidized, it fluoresces. So if we have the respiratory burst, we're producing lots of oxygen reactive species, and therefore we are oxidizing dihydroxyrhodamine. And so this is what you see here for a control person. So we stimulate the neutrophils for them to produce these reactive oxygen species, and if they function, they will oxidize dihydroxyrhodamine, and that's what we see here. And then if you do the same thing with the neutrophils of a patient before it's treated, you can see that this process is much less efficient. There's a lot of dihydroxyrhodamine that doesn't fluoresce properly, therefore it's not being properly oxidized, and that's because we are not producing enough NADPH for the NADPH oxidase to work. And then you treat the patient, and you do the same experiment with their neutrophils, and you see that again you have recovered most of the ability to do the respiratory burst. So we had neutrophils that even though they were not free of anhydroglycitol 6-phosphate, they were now functioning much better. And so the last thing we looked at was the neutrophil counts. So this shows you the neutrophil counts in this patient. So before being treated with empagliflozin, this patient was taking this GCSF. And so what you see is that, so you get injections, and then when you get an injection, the numbers increase of neutrophils. And then because the neutrophils of this patient were still full of anhydroglycityl 6-phosphate, they were dying very quickly. Okay, so when we started the treatment, the patient stopped taking GCSF, and you could see that indeed the neutrophil counts went down to almost very close to zero, and they started taking, we don't do that anymore. Usually if they're taking GCSF, we keep the GCSF, and then they start on empagliflozin, and then we only stop the GCSF later. But nevertheless, this is what we did here. And so the neutrophil counts dropped. And then after, once we had decreased the amount of anhydroglycityl and probably the amount of anhydroglycityl 6-phosphate, then the neutrophil counts began to increase. And then in this patient, they reached numbers which were 600, 700. So this patient remained neutropenic. Some patients, they go up to 2000, maybe even more, it depends. The G6PC3 patients, they reached higher numbers. But nevertheless, this was sufficient for there to be a huge clinical improvement in this patient, because you could see that the patient told us that for the first time she could brush her teeth without pain. Her stool frequency went down to 2-4 times a day, which is much better than what she had before. Her weight increased by 2.5 kilos, and that's probably because her intestine was in much better shape, so she reabsorbed more nutrients, etc. She could stop the GCSF injections. So her neutrophil counts remained low, but nevertheless, she had functional neutrophils, and she didn't really have any hypoglycemias. So I'm getting to the end of my talk, and I'd just like to talk about one or two more things. So I told you about this anhydroglycityl, and I'd like to tell you where does this anhydroglycityl that it's present in food come from. So again, here it's also a matter of a mistake or a side reaction, if you like. And so usually when we have complex sugars like glycans, glycogen, starch, whatever, oligosaccharides, so these are hydrolyzed to give monosaccharides and glucose. And so it's a hydrolysis reaction. And then here you can make glucose. And these are glucosidases that do this kind of thing. So some of these glucosidases that are in our intestine, or maybe they are in the bacteria that are in our intestine, or in the bacteria of the cows that produce that have the meat that we eat, or wherever, or in the vegetables, or etc. So they will act on these complex carbohydrates, and sometimes some of them have a side reaction, and instead of having a hydrolysis and taking in a molecule of water, they have an elimination reaction, which is a minor reaction, and it leads to the production of 1,5-anhydrofructose. Now, we have in our cells an aldehyde reductase, which is very efficient to convert 1,5-anhydrofructose to 1,5-anhydroglycityl. And so this is the origin of this 1,5-anhydroglycityl that we have in the body. So, this is a slide that I made where I wanted to show you all the possible sites of lack of specificity or side activity of enzymes, or lack of specificity of transporters that leads to the production of this anhydroglycityl 6-phosphate and the toxicity of the anhydroglycityl 6-phosphate, so that we have to have this repair system to allow us to cope with all these mistakes or the presence of this side activities that we have. So I've told you that the glucosidases, they are not specific to produce glucose. They would sometimes produce 1,5-anhydroglycityl in our intestine. This 1,5-anhydroglycityl ends up in the blood. And so it stays in the blood, because in the kidney, we have a transporter that is going to reabsorb 1,5-anhydroglycitol back into the circulation and prevent it from being excreted in the urine. So we think this is SGLT5. We think the role of this transport is not to transport anhydroglycitol, but we don't know yet. But we're studying it a little bit now. But nevertheless, it's a lack of specificity of this transporter, so the second lack of specificity, that is making us keeping 1,5-anhydroglycitol in the blood. Then 1,5-anhydroglycitol, because it's in the blood, gets into the neutrophils, probably by the same transporter. We don't really know yet. And here it's the side activity. So the third mistake, the side activity of the exokinases, which are there to phosphorylate glucose into glucose 6-phosphate, that are going to act on 1,5-anhydroglycitol 6-phosphate. And then the fact that 1,5-anhydroglycitol 6-phosphate becomes a problem, it's because it binds to the binding site of exokinases so the exokinases have this binding site for glucose 6-phosphate that regulates its activity in the cell. And so because anhydroglycitol 6-phosphate looks so much like glucose 6-phosphate, it's going to bind to this inhibitory binding site, and it's going to block the exokinases. So you have here 4 lack of specificity, side activities, whatever that make that we end up having to have this repair system to prevent the toxicity of 1,5-anhydroglycitol 6-phosphate. So now I just like this, this is the things that I'd like you to remember. And then I have one more slide and then I'm done. So, so the take-home message would be G6PT and G6PC3 collaborate to destroy the non-classical metabolite 1,5-anhydroglycitol 6-phosphate. The accumulation of 1,5-anhydroglycitol 6-phosphate in neutrophils causes neutropenia by blocking glucose phosphorylation. Okay, so when we deplete the blood, when you deplete blood of 1,5-anhydroglycitol with Sglt2 inhibitors, we treat the neutropenia in the G6PC3 deficient mice. And then if we use the Sglt2 inhibitor therapy in the patients, we reduce 1,5-anhydroglycitol in plasma, 1,5-anhydroglycitol 6-phosphate in leukocytes, we increase neutrophil numbers, improve neutrophil function, and this leads to an improvement of patients' clinical outcome. And then last but not least, neutropenia in GSD1B and G6PC3 deficiency joins the growing list of inborn errors of metabolite repair. And so I have one last slide here, just to show you a series of abnormal metabolites that we have somehow worked here in the lab and that are well known for us here in the lab. There are others that others have discovered, and they're not all here, but this is just a series of them. So we've spoken about L2-hydroxyglutarate, and we've spoken about 1,5-anhydroglycitol 6-phosphate, who then leads to neutropenia. And so this is also, these 2 metabolites are also quite interesting. It's 4-phosphoerythronate, which is made by a side activity of GAPDH on erythrose 4-phosphate. And then you have 2-phospholactate, which is made by a side activity of pyruvate kinase on lactate. So when these 2 abnormal metabolites accumulate, they are going to inhibit 4-phosphoerythronate, will inhibit 6-phosphogluconate dehydrogenase, and the inhibition constant is below 1 micromolar, so very potent inhibitor. And 2-phospholactate is going to inhibit PFK2, the enzymes that make fructose 2,6-bisphosphate, one of the well-known regulators of glycolysis. Now, there are no diseases associated with the lack of the repair enzyme, which is phosphoglycolate phosphatase, and probably because it is incompatible with life, because when you do the mouse knockout, you see that the mice are embryonically lethal. So these are abnormal metabolites that are related to carbon metabolism, if you like. So this is another type of abnormal metabolite to do with cofactors. So here, the abnormal metabolite is a hydrated form of NADPH or a hydrated form of NADH, which we called NADPHX or NADHX. So when these abnormal metabolites accumulate, because we have a deficiency in one of these two enzymes, so an epimerase or a dehydratase, these will inhibit a series of dehydrogenases and lead to a neurological disease. And so the other type of metabolite, the last one, it's related to amino acid metabolism, if you like, because it's a form of glutathione which is deaminated. So what's happening is that there are various transaminases that will deaminate glutathione, will work on glutathione, and they will deaminate it. And then when you don't have the amidase that is able to destroy this deaminated form of glutathione, which is NIT1, then deaminated glutathione accumulates. So we don't really know why this is a problem. So what it is inhibiting in the metabolism, probably enzymes that use glutathione. But what we now know is that there are at least 3 families, I think, that have a mutation in the repair enzyme, and that in this family you find patients that have neurological psychiatric disorders. So this gives you an idea of the extent of these abnormal metabolites, where they can be formed by a series of various enzymes. And it probably tells you that this is just the tip of the iceberg. We just looked here at some enzymes that are able to have side reactions, but I mean, there's no reason why there won't be many others that would also have side reactions and that would make these abnormal metabolites, for which we would need repair enzymes that we don't know yet what they are and that we probably need to find, and which might explain some of these inborn errors of metabolism. And I've reached the end, so I just like to thank all the people that allowed me to tell you what I've just told you today. So of course, the main person is Professor Emil van Schaftingen. So we've worked together for about 30 years. We told together many metabolism related stories. This is for me probably the most beautiful one. But so, I mean, you know, all I've learned from him. So he's really my mentor and somebody I have a lot of respect for. These are the people in the lab. Guido Bomer has his own group, but he's the one who worked most on the phosphoglycolate phosphatase. Alessio Baracchi worked with me on the NIT1. Then Carole Inster in the University of Luxembourg worked mainly on the NADPH X story. And then these are many of the clinicians that work with me, you know, that have patients and that we've been treating together. And then I'd like to thank you for listening to me, and I'm ready to take questions.
Thank you, Professor Cunha. That's very informative. I love metabolism. And I also like these stories, how they intersect with diseases. As it's customary, Arpana Vania, our academic coordinator, will now lead the discussion. Arpana.
Great, thanks, Oliver. Wonderful talk, Maria. We have a few questions in the chat. We'll just start right away. There's a question about why you did not test galactose-6-phosphate and glucose-1-phosphate as substrates for G6PC3.
So galactose-6-phosphate and?
Glucose-1-phosphate. Oh,
I did test glucose-1-phosphate, so that was not a good substrate. I don't know if I tested galactose-6-phosphate. I guess galactose. Yeah, I did test glucose-1-phosphate and fructose-1-phosphate. I mean, I tested a series more, and I just put there the best ones.
Okay.
Yeah, I don't know if I tested galactose-6-phosphate. I don't know.
There's a question more in regards to type 2 diabetes. And it's kind of like the chicken and the egg question. What happens first? In type 2 diabetes, does it lead to high 1,5 Ag, or does type 2 diabetes, is it a consequence of abnormal 1,5 Ag?
No, so 1,5 Ag, we all have it. We don't have to be diabetic to have 1,5 Ag. You have it, I have, everybody has. And so we all make 1,5-nitroglycerol 6-phosphate. We all make it, but we have the repair system, so it doesn't accumulate. It's not a problem for us.
Okay.
But if the patients don't have either the transporter or G6PC3, it then becomes a problem. Now, in type 2 diabetes, what is happening is that you have glucosuria, you see, if you're not treated. And so if you have glucosuria, you won't be able to absorb from the renal filtrate the 1,5-nitroglycerol back into the circulation. And so that's what's happening.
I see. Oliver, you had a comment.
Even if you don't take the Sglt2 inhibitor.
Mm-hmm.
Well, usually 1,5 Ag is in biomarker or higher levels of blood. 1,5 Ag is a biomarker for diabetes. So
yes, because if you're diabetic, so if you're if you're diabetic, you will have less 1,5 Ag in your blood. Because you have, yes, because you have glucose in the urine.
Yeah, I hear you. I remember differently, but I haven't read about it for a while.
Yeah, no, but it's like that. So it's actually a way. So if the patients are taking their anti-diabetics and are being treated well, their level of 1,5 Ag would be normal, like yours and mine, because they don't have glucose in the urine. If they are not taking well their drugs and whatever, they'll have glucosuria, and so their 1,5 Ag would lower in the blood. And that's why it's a marker for diabetes.
Okay, that makes sense. Yeah, I liked the figure that you showed in terms of like blocking, having the receptors open and blocking that kind of helped me understand what was going on. Next question, there was a question on more about your methodology on how you extract your leukocytes. Do you use the Buffy coat for leukocytes? If so, how much of the Buffy coat do you need to use to measure 1,5 Ag and other metabolites?
So we actually use, we use this gradient, which is called lymphoprep. So what we do is we centrifuge the blood, the whole blood on this gradient, and then we have, we can like that isolate the lymphocytes. So we have a PBMC ring and we take them out, and then we have a pellet where we have red blood cells and neutrophils. And so we lyse the red blood cells and we recover the neutrophils. Okay, and then we make an extract that we use at the GCMS and you can do that with 1 ml of blood is more than enough. And so we even do it in a slightly easier way because red blood cells, they don't accumulate nitroglycerol 6-phosphate for some reason that we don't understand very well. Probably either there is another phosphatase or it's not transported, but very surprisingly, red blood cells don't accumulate. So we can just prepare an extract of whole blood and measure accumulation of nitroglycerol 6-phosphate like that. And for that, we use 400 microliters of blood, and it's enough. And so we do methanol chloroform, and then we can put that at the LCMS, and we can see that.
Okay. Thank you. Going back to 1,5 Ag, can you explain again how 1,5 Ag competes with glucose for G6PC3? And in different tissues, if it has both of those metabolites, what is better substrate for G6PC3? I guess she was really specifically asking what is the Km for each of those metabolites?
So G6PC3 has, and I'm not sure I remember exactly, but the Km for glucose 6-phosphate is much higher. So G6PC3, it dephosphorylates a little bit glucose 6-phosphate, but it's not very good. I mean, we have another, we have another enzyme that do that. And on the top of that, so to dephosphorylate glucose 6-phosphate makes a lot of sense in the liver, right? Because we have glycogen, we need to produce glucose. But in the neutrophils, to have a G6PC3, so an enzyme that acts a little bit on glucose 6-phosphate, I mean, neutrophils don't serve to produce glucose. So it's there. Yeah, it's there obviously for another reason. And it's there to dephosphorylate 1,5-hydroglycityl 6-phosphate. So it's the Km, I don't remember exactly what is the Km, but in the PNAS paper, there is a table that has all this information. But I think the Km for nitroglycityl 6-phosphate is better than is the Km for glucose 6-phosphate. And the activity is certainly better.
Okay, I hope that addressed that question.
The amount, and the amount that we have in the cell, it's much higher, the amount of nitroglycityl 6-phosphate.
Okay. When thinking about the population, is there data profiling the differences in these enzymes specific to different types of population or just in general? Do you think the differential expression or functions of some of these enzymes could explain some immune system disorders?
Of the G6PC3 and the G6PT?
Yeah, I think so. Yes.
Actually, so if you're heterozygous for this, you're fine. So these are kind of, so you're born like that. So you have to have, both copies of your gene have to be mutated. So you really have to be very deficient for there to be a problem. So I don't think it's related to variations in the expression of these two proteins, the transporter and G6PC3.
Okay. And then last, one of the other questions that came up just right now, when you mentioned just seeing the tip of the iceberg for repair mechanisms, a lot of work has gone into discovering that there was an issue with metabolite repair in the first place. Do you feel like there could be a more untargeted way to mine out error and repair relationships, allowing us to see the rest of the iceberg, or, to see what's underneath?
The difficulty is that if you take normal cell, you don't see these abnormal metabolites because the repair enzymes are there, right? So unless you come with a hypothesis and you can delete the repair enzyme, there you can see the accumulation. And even in the case of G6, in the case of G6PC3 and G6PT, when we incubated the cells that were deficient in G6PC3 or G6PT, in the absence of nitroglycityl, we couldn't see anything. So maybe in some cases, but often I think you have to come with a kind of hypothesis in order to make the good tools, which means cell models without a repair enzyme that, you think that you see when you study enzymes, you see that there is a side reaction in vitro, and then you can start thinking, oh, can this side reaction happen in vivo? And then imagine, if it's a phosphoric ester, imagine a phosphatase that it's expressed in the same tissues as the enzyme that is doing the side reaction is, and then, do cell lines where you knock out potential phosphatase and then put it in the LCMS and then see, yes, can I now see the accumulation of, of the abnormal product. So, yeah, I mean, we never found anything using N-targets. Let's put it this way. We never found anything using N-target metabolomics.
Yeah, that seems like there's a lot of trial and error leading up to, you know, analyzing it in the GC or the LC. I had a question, maybe kind of going back to the bigger picture. What was the motivation to investigate neutropenia? Like, I really don't know what it is. Is it specific in certain groups, or, you know, is it a...
Neutropenia is when you have too few neutrophils in your blood. So if you have too few neutrophils, you have infections.
Mm-hmm.
You see, and what was weird, and that's why we started to work on it, what was weird is that the glucose 6-phosphate transporter in the liver, it makes a lot of sense, okay? Because it is providing glucose 6-phosphate to the glucose, to the glucose 6-phosphatase to produce glucose so that we don't have hypoglycemia when we fast. So this was very logic. So if you don't have the transporter, you have a big liver full of glycogen because you cannot degrade the glycogen, you have hypoglycemia. If you don't have the phosphatase, exactly the same thing happens. And then this very bizarre thing, when you don't have the transporter, you also have neutropenia, which is very surprising. And then much later on came this new disease where you find that the homologue of the glucose 6-phosphatase, okay, is mutated. And these patients, they only have neutropenia. They don't have any problem with the glucose metabolism. And so what people kept saying in the literature, it's to do with the transport and the hydrolysis of glucose 6-phosphate because G6PC3 had a little bit of activity on glucose 6-phosphate, and because the transporter was transporting glucose 6-phosphate. And then, you know, you could see the mice were neutropenic, and then, but for us, this thing just didn't make sense. There was no logic of having a homologue of G6PC1 that would also serve to dephosphorylate glucose 6-phosphate. That's how we came up with, there must be something else.
Mm-hmm.
And then we went and looked for other phosphoric esters, which would be substrates of G6PC3. So that's the logic that we have.
Interesting. Yeah. So all the chat questions are done. We have a few minutes left if there's any, you know, one who wants to.
Thank you. Yes, thank you, Veiga, Cunha. That was really interesting. It's always delightful to see actual biochemistry and not just patterns and statistics and trend analyses, but rather mechanisms, especially to think about transporters and organelles and not just, you know, enzymes in a bag type of views of biochemistry. I love it. So thank you again, Maria.
Thank you. Oh, thank you.